Thanks for coming out. I'm Michelle McCauley. I'm with Levi Moran at Emory University, and I'm going to tell you about an empirical test of Baker's Law. But first, I'd like to acknowledge that this project started as an undergrad honors thesis by Will Milligan and has passed through a lot of people before it finally got to me. What if we advance the slides? Okay. So one thing that has always fascinated me is the incredible diversity we see in mating systems in nature. We see diversity within species and then, or among species and then within species. So for example, all of these photos are examples of what we would call mixed maters, which are organisms whose reproductive system allows for either outcrossing or self-fertilization. And then additionally, in all of these examples, we see a lot of variation between populations in rates of self-fertilization. So if we look at our friend, the hairy tomato, this is a map of the hairy tomato's native range in South America. And in this paper by Markova et al., they sort of went into these different populations and they scored them for their self-compatibility rates. So in the white triangles, these populations are largely self-incompatible or obligately outcrossing, whereas in these orange squares, these populations are mostly self-compatible or capable of self-fertilization. And you can see here that there's a pretty clear pattern where our obligately outcrossing populations are at the sort of center of the species range, whereas the self-capable populations are more towards the edge. And this is a pretty common pattern in plants, although not universal, that we see more self-fertilization at the edges of species ranges. And so this pattern is called Baker's Law. So what is Baker's Law? Well, it's not a law. Baker's Law is a type of pattern that was observed by the botanist Herbert Baker of an enrichment for the capacity to self-fertilize in more recently colonized areas compared to the species range core. And Baker's hypothesis for what was driving this pattern was that long-distance dispersal will select for uniparental reproduction, so self-fertilization or asexual reproduction, due to reproductive assurance in the face of mate limitation. So when I say long-distance dispersal, I'm not talking about like a specific distance. I just mean how far you need to go so that you no longer have readily accessible mates. So bringing it back to our hairy tomato, Baker's hypothesis here would be that we see more self-fertilization on the species range edges because these areas have been more recently colonized. So in order to kind of dig in more into how that would work while also simultaneously introducing my study system, I'm going to start talking about C. elegans nematode worms. So C. elegans, like many plants, has a mixed mating system. C. elegans are androdioecious, which means individuals are either males or hermaphrodites, and hermaphrodites are able to self-fertilize or outcross to males. Although I should note, under standard lab conditions, C. elegans are primarily self-fertilizing. C. elegans are a fantastic lab organism. They eat bacteria and can be readily grown just on a petri dish on a lawn of E. coli. My cartoon is not to scale. Adult C. elegans worms are approximately 1 millimeter long, which means we can grow entire populations in a single dish. So C. elegans is a dispersing species with a boom-bust life cycle in nature. I know, nobody ever talks about what they do in nature. So to explain my incredible PowerPoint art, this is meant to represent a rotting piece of fruit. These sort of yellow-orange clouds are meant to be bacteria on this apple. So worms will colonize this fruit and eat said bacteria. So while there's a lot of bacteria available, they'll progress normally through their life cycle and their populations will rapidly expand. However, once they've exhausted their food source, signals from crowding and starvation will cause larval worms to develop into an alternative life stage called dower. And dower worms are physiologically and behaviorally different. These worms grow really long and skinny, they stop eating, and they become highly motile. They'll also sort of like stand up and wave around in hopes of hitching a ride on a friendly invertebrate. And so via voracy, these worms are able to move incredible distances, although I also want to note dower worms are very accomplished crawlers, so they can get pretty far even if they can't hitch a ride. So worms will sort of like cruise around until they find a new food source, such as another rotting piece of fruit. The worm will develop out of dower and progress into its adult life stage. So worms coming out of dower actually have a higher propensity to outcross. So once this worm is an adult, it will outcross at higher rates if there are other mates available. However, if you just hitched a ride on a random invertebrate and just showed up in the next place where you found food, you might not have another suitable mate available. But luckily for this worm, it's a hermaphrodite, no big deal. The worm can just self-fertilize and establish a new population via self-fertilization. So this is what I mean when I say that the ability to self-fertilize confers reproductive assurance in the face of mate limitation. So you might be thinking, okay, that makes sense, but is there any evidence for it? So most studies of Baker's law are done in plants, although Baker himself noted that this pattern and the underlying processes could also happen in animals. They're mostly correlative studies with natural populations, and because natural populations have a lot of things going on in them, results tend to be either mixed or ambiguous, and there have been very few direct or experimental tests. So in our lab, we asked, in our worms, does repeated dispersal select for selfing? And we answered this with an evolution experiment. So in our evolution experiment, all of our starting populations are 95% worms that have been genetically manipulated to be obligate outcrossers. And to this, we added a small number of mixed mating worms capable of self-fertilization, and both subpopulations have a shared genetic background. And this setup is meant to mimic that hypothetical I described earlier, where a mostly obligately outcrossing population has a few individuals that are self-compatible. So then we exposed our worms to our different treatments for 12 rounds of selection or passages, which is our x-axis, and then we monitored their selfing rates over time, which is our y-axis. So we had a no dispersal treatment and a dispersal treatment. We induced dispersal by spatially arranging the bacterial food sources on a plate. So each of these big circles is meant to represent a petri dish, and the yellow circles within it are our bacterial patches. So in our no dispersal treatment, all the patches are around the center of the plate, whereas in dispersal, the patches are around the edges of the plate, and we placed our worms in the centers indicated by these red stars. And the worms had to be able to move to their food source in order to grow and reproduce. So we gave these worms time to disperse, and then we selected one bacterial patch at random, and the worms on that patch were moved on to the next dispersal plate into the center. So some notes, worms distribute more or less evenly between the 4 patches, although on our no dispersal plate, because the patches are close together, we saw a lot of movement of worms between patches, so those worms are seeing more potential mates, whereas on our dispersal plate, we didn't see very much movement at all between patches, so those worms are more mate limited. And then in my ancestral populations, I found no evidence of inbreeding depression. So in addition to dispersal and no dispersal, we also had worms that were in dower versus worms that we did not induce dower in. So I'm going to look at the results for the no dower worms first. And we see that selfing rates increase in both our dispersal and no dispersal treatments, although in the dispersal treatment, selfing rates increase much more quickly. But when we look at our dower worms, in our no dispersal treatment in green here, we don't see a significant change in selfing rate over the course of the experiment. Meanwhile, in our dispersal treatment, we do see selfing rates increase. And given that dower is the life stage in which worms disperse in nature, this is the more ecologically relevant experiment. So this is a pretty powerful result that we were able to see this difference. Okay, so then we wanted to dig a little bit more into the dynamics of why we were seeing this, so I'm going to show you some results from fecundity assays. So we had worms from our two different subpopulations, obligate outcrossers OO and mixed maters MM. The mixed maters are primarily self-fertilizing, obligate outcrossers outcross. So we then put them either on dispersal or no dispersal plates for 20 hours, and then counted the total number of worms, the total number of females or hermaphrodites, and the total number of eggs. And 20 hours is not the full period over which worms lay eggs, but when you're in a food-limited environment, how quickly you can reproduce is a really crucial. Okay, so when we look at our no dispersal treatment, we see that our obligately outcrossing females have a much higher average fecundity than our mixed mating hermaphrodites. However, when we look at the dispersal treatment, this difference disappears entirely, and we see no difference between them. So in a less mate-limited environment, such as the no dispersal plates, we do see that our obligately outcrossing females do experience some sort of fecundity advantage within the first 20 hours. But this is only per what I've eloquently called egg-producing worm, so hermaphrodites are females. When we look at a more population level, yeah, so I'm now going to show you these are the same assays, but it's egg per worm. This includes the males that exist within the population. So when we look at our no dispersal treatments, we now see that they're on the level of egg per worm. There is no difference between our mixed maters and our obligate outcrossers, and this is likely because males do not directly contribute to the production of offspring. But when we look at our dispersal plates, we now see that we have more eggs per worm in our mixed maters, so our selfing hermaphrodites, versus our obligate outcrossers. So the overall takeaway from this slide is that outcrossing females do have a fecundity advantage when their access to mates is high, but we still see an advantage on the population level to selfing under dispersal. Okay, so my conclusions. So we have experimentally supported that dispersal can favor selfing over outcrossing. And I wanted to note that we only explored the scenario of repeat dispersal. Baker's law could still apply under other circumstances, such as range expansion or invasions. And I also wanted to note that I am in no way arguing that this is the only driver of the patterns we see of selfing and outcrossing in nature, only that it could be a contributing factor. And then finally, depending on who's in this audience, this might be kind of preaching to the choir. But if you're like me and you just work in a lab with a model system, this is the difference between our dower and our no dower worms is sort of a call to keep your model system's ecology in mind when designing experiments. So I'd like to thank all of you for listening, and I guess.
Sorry about that, everybody. Thanks for coming. My name is Daniela Wilner. I'm at the University of New South Wales in Australia. I'm going to tell you a little bit about what I've done in my PhD so far. So yeah, let's get started. Let's talk about the birds and the bees. And also all of the other animals that reproduce sexually, because there's so many of them, and it's actually really bizarre, right? We've been wondering about this for over a century now as biologists, because sex is quite an inefficient and costly way to reproduce. Not only is there the famous twofold cost of sex, but there's also all sorts of other risks and costs associated with mating that females have to face if they're going to do that. Now, asexual reproduction comes with its own set of limitations, especially in the long term, but theory predicts that a flexible or facultative strategy should be optimal. And so animals that can do both, so that can reproduce both sexually and asexually, should be really common. But they're not, except C. elegans, which, wow, that was so cool. Also, I don't know, we'll need to chat later, but a lot of these animals that can reproduce both ways occur in weird geographic mosaics, where some populations are made up entirely of females reproducing asexually, and others are mixed sex and have a whole lot of sex. So it seems like in each of those populations, something is tipping the balance towards one of the two obligate strategies, either sex or asexuality. And if we can understand what that is, it might help us understand how these different reproductive modes evolved in the first place. So that's why I'm studying one of these animals, the lovely peppermint stick insect. These guys live in far north Queensland and northern Australia in the tropical rainforest. And they are facultative parthenogens, meaning the females can reproduce both sexually and asexually. So if eggs are fertilized, they develop into an even proportion of sons and daughters. And if they're not fertilized, they still hatch just fine, and they only produce daughters. So the females can switch flexibly between the two modes. And despite this flexibility, they occur in one of these strange mosaics I was telling you about, where some populations are made up entirely of females, all reproducing asexually, and then the others have an even proportion of males and females, and the males are almost constantly guarding and mating with the females. And these populations weirdly occur really close to each other. So I wanted to know what's maintaining these distinct sexual and asexual populations so near each other. And I hypothesized that it has something to do with female resistance. So maybe some of these females are resistant to sex, and that's why those populations are remaining male-free. Now, my lab mate Soleil found that sorry, did I mention this was in Australia? Yes. So my lab mate Soleil found that the populations in Australia all fall into two major phylogenetic clusters. One that contains all the populations in the north of the range, and one contains all the populations in the south of the range. And that northern genotype or cluster contains mostly mixed-sex populations, while the southern genotype or cluster contains mostly all-female populations. So I decided to bring females from each population type into the lab and cross them with males so that I could count up how many sons and daughters they produce to estimate how many eggs were fertilized and see if there was a difference between those two different kinds of females. So here are the results. We're going to have female type on the x-axis, so females either descended from southern all-female populations or northern mixed-sex populations. And remember, these were all paired with males, so they all had a chance to mate. And on the y-axis, we're going to have the offspring sex ratio, so the proportion of sons. So when that's 0, it means they didn't produce a single son. And if it's 0.5, it's an even proportion of sons and daughters, which is what we expect when eggs are fertilized. And what we found is what I expected based on my hypothesis. So the females descended from all female populations produced very few sons, which suggests that very few of their eggs were fertilized. So that was pretty exciting. And remember how I told you that these come from two genetic clusters? So it's tempting to say, ooh, this is a genetic effect, right? But we have to keep in mind there's also room for maternal effects here, because females from all female populations are necessarily impatternate, meaning they had no fathers because there's no males in those populations. Whereas females from mixed-sex populations are most likely patternate because most of their mothers would have mated. And I wanted to control for that. So I actually used the second generation of lab-reared insects in this experiment, and I manipulated whether the mothers of these females had mated. And so in this way, I could have both patternate and impatternate females from those mixed-sex populations. And those impatternate females from the mixed-sex populations produced an intermediate offspring sex ratio. So that suggests that there's both a genetic effect and a maternal effect affecting this resistance. Really cool. But more questions. First of all, what is this female resistance? Is this something behavioral that happens, some resistance to mating, or is it something that happens after mating? And secondly, could this actually prevent males from invading natural all-female populations? Because a couple insects in a tiny container in the lab is a far cry from wild populations in the rainforest. So I couldn't exactly go out into the rainforest and start introducing males into new populations, might be a little frowned upon. So I did the next best thing. I set up semi-natural enclosure rooms at the university in controlled temperature rooms, and I put 5 large host plants and 4 females into each room to create a miniature all-female population. And I did that with the 3 different kinds of females we've been talking about, so that I could create 3 different kinds of miniature populations. And then once the females had time to acclimate to their new home, I introduced a male, and I let them do their thing for 2 weeks to see what would happen. But when I first introduced the male, I observed their behavior for the first hour. So I kept track of whether that male left his plant in search of a female and whether he managed to actually mate in that first hour. And then I kept checking up on them for the next 2 weeks. So twice per day I came in and looked at who was with who was where, and whether the males were mating or guarding any of the females. And finally, when the 14 days were up, I wanted to look at the outcomes. So I took everyone out of these semi-natural enclosures, and I put each female in an individual container so I could collect her eggs, and again, count the sons and daughters to estimate fertilization success. And I did that a bunch of times. So I did at least 6 rooms per female type. And I'm going to show you some preliminary results now. So again, on the x-axis, we have female type. So these are either populations made up of impatternate females from all female populations, impatternate females descended from mixed-sex populations, or patternate females descended from mixed-sex populations. Mouthful. And on the y-axis, first we're going to look at what happened in that first hour. So it's going to be the proportion of males that did the different behaviors. And you can see there's a qualitative difference here. So when the room had females from descended from all female populations, most of the males didn't do anything. Only one of them even bothered to leave his plant, and he didn't even try to mate with anyone. Whereas if the females were descended from mixed-sex populations, I saw a lot more searching behavior in pink and more mating, especially if the females were patternate. But that's just the first hour, right? Like what happened later. So this is going to be the percentage of females in each enclosure that I saw mate over the next 2 weeks. And the pattern holds. So you can see if the females were from all female populations, the males had very poor reproductive success, or mating success. And the highest mating success was with the patternate mixed-sex population females. And finally, we can look at the outcomes. Did this actually result in a difference in fertilization success? Yes. So the females from all female populations produced, again, very few sons. So all those dots along the bottom are females that didn't produce a single son. And further, we can combine this with the behavior data. We can focus in on the females that I definitely saw mate, and we can see that there's still an effect. So even, these are females that I definitely saw mating at least once, and you can see that even then, if they were from all female populations, they didn't produce many sons, suggesting few eggs were fertilized. So altogether, this suggests that yes, the females from those all-female populations are resistant to mating and fertilization. And this is due both to a genetic effect and possibly a small maternal effect. And secondly, it's driven by both pre- and post-copulatory mechanisms, because we saw that the females were less likely to mate, but also less likely to have their eggs fertilized after they mated. And yay, it held in a more natural environment. So I saw the same patterns in the tiny containers as in the semi-natural enclosures. So you could imagine how in the wild population, if a male were to wander into one of these small isolated all-female populations, this resistance would be able to impede male invasion, because that male would have very low mating success. Even if he mated, he'd have low fertilization success, produce very few sons, and then those sons would face the exact same obstacles. And if female resistance does, in fact, promote the maintenance of all female populations in this way, it could help form these weird mosaics. And over time, it might lead to obligate asexuality. So for my next steps, I'm trying to look into the mechanisms of this resistance. So those weird brown things over there are the female sperm storage organs. I'm trying to see if there's a difference. The cartoon sperm are not to scale. And I'm also looking into bacteria, because there's bacteria that are known for manipulating reproduction. So stay tuned. I'm sure we'll find some cool stuff. I actually just got the sequences today, so I'm really excited. But yeah, thank you all for listening. Thank you to my supervisors and my amazing lab mates and office mates and all my collaborators, all the people that helped me throughout my PhD, and especially my amazing volunteers who helped me rear so many insects. This would not have been possible without them. So thanks, and I'm happy to take any questions. Yes?
What's happening with voiding number?
Short answer, we have no idea because we haven't been able to successfully look at their chromosomes. We do know their heterozygosity drops. My lab mate Soleil's done basically a whole PhD on that stuff. I think you were first.
If you took like a female from the female population and you like made it mate, how many generations do you think you would get before you had the effect of the intermediate one?
So I actually did do that. In that experiment that I showed you guys, I only showed you the 3 kinds of females, but I also had, because I used the second generation, I had the daughters of the females from all female populations that mated with the males. And those acted pretty much like northern mixed-sex population females.
And then one more question. Will the female move away from the male, or is it like a pheromone thing that's like, oh, don't even come near me, don't even think about it?
That's an excellent question. I think it's both. So one of my lab mates actually did a small pheromone experiment, and it was pretty weird results, but it seemed like the males were less happy to move around when they were So yes, I think it's both, but I don't know.
So thanks for coming to my talk, everyone. My name's Kate O'Hara. I'm a PhD candidate from the Australian National University in Canberra, and I'm also doing my PhD in conjunction with Australia's National Science Agency, the CSIRO. So we've got another Australian parthenogen, yes, represent. But in this case, we can see we have reptile system, and for all you evolutionary biologists, we have a number of features that make this system quite interesting to study. They're hybrid, they're triploid, and they're asexual. Interesting, but also really annoying, as I've found out. Is this going to go? Oh, yeah. But first, I don't really need to do much of an introduction on parthenogenesis at this point because Danielle's done a wonderful job for me. But it's probably something you've heard about a lot in the media, even if you don't directly study it yourself. There have been a whole suite of reports of animals that have been able to reproduce without having sex. I've been asked to talk on the radio about some of these, and I'm like, well, I don't study that sort of parthenogenesis. Of course, that is a facultative parthenogenesis, but that's not what my system is. Now, parthenogenesis, we hear it thrown around. It comes from the Greek, meaning virgin birth. Sounds a lot like the Parthenon, because that is the house of virgins. That was something interesting that I found out during this. And yes, so facultative pathogens can swap between those asexual and sexual reproductive modes. But what I'm interested in are obligate pathogens, and in particular, the vertebrates, because there are a number of insects that do this, but vertebrate systems are slightly different, partly because they have much smaller population sizes, and that's going to affect some of their strategies and their genetics. So some notable examples of obligate vertebrate pathogens include the whiptail lizards, Aspergillus, the Amazon molly, and of course, Heteronodia binoi, or the prickly gecko, which you're going to hear a lot about in this talk. Now, as we've already heard, these asexual vertebrates are exceptionally rare. They make up only 0.1% of all vertebrate species on the planet. And, well, why? As you've heard, there are many advantages to being asexual. You don't need to find a mate. You can, there's the twofold demographic advantage, as every single individual can bear offspring. Your population can grow quite twice as quickly, but they're so rare. So over the years, evolutionary biologists have proposed a number of theories to explain this rarity, and most of them hinge around the idea of reduced genetic diversity. Of course, if an organism is clonal, it's lacking the diversity it might need to adapt to a rapidly changing environment. It doesn't have sex, it doesn't recombine, it's relying purely on standing genetic variation and then subsequent selection on clones to adapt. So that probably explains their rarity. That said, there is this 0.1% of asexual vertebrates that we see alive today. They're probably a small subset of all asexual vertebrates that have existed on Earth. But what makes them special? So if we look at them a little bit more closely, there tend to be a few correlates of vertebrate parthenogenesis. They're typically of hybrid origin, so they're the result of two separate species coming together at some point in time in the past, and they often have elevated ploidy. So this is a karyotype from a heteronodia binoid pathogen, and we can see that it is a triploid. Population genetics nightmare, but also potentially a way that it can generate more genetic diversity than if it only had 2 copies of its genome. So that has led evolutionary biologists to think that rather than a lack of genetic diversity explaining their rarity, since they have these really complex genomes, perhaps it's just really, really hard to form vertebrate pathogens in the first place, because if they're going to be successful and they need to be a hybrid and they need to have this extra chromosome copy, how often does that actually happen? So you've got to slightly disrupt meiosis, but not disrupt it so much that you end up with inviable offspring. So there's a whole suite of papers on things called the balance hypothesis or the rare formation hypothesis, which looks into what are the conditions required to form pathogens. But I'm not going to go into that anymore now. That's not a focus in my PhD. I'm interested in looking at what is the genomic landscape in pathogens that are extant today. So in particular, the first chapter of my PhD, I was asking to what extent do these heteronodia binoid pathogens display genotypic or genetic diversity, and how is this influenced by their hybrid origins and their polyploidy? So just a bit of background on the system. So this parthenogenetic system was actually discovered in the 1980s by my supervisor, Craig Moritz. It was not meant to be the focus of his PhD, but it was so interesting that I think he got 4 papers out of it. But since the 80s and the 90s, there hasn't really been any modern genetics work done on the system. But from karyotype work, we do understand a little bit about their history. So they are the result of a hybridization event, 2 hybridization events actually, that have occurred hundreds of thousands of years ago, so they're not particularly old, as most pathogens are. And these are the 2 parental lineages. Heteronodia binoid is a species complex, and they're being given the very memorable names of CA6 and SM6, which you don't need to remember. So we're just going to think of them as the AA diploid and the BB diploid. So they've come together, they've mated, they've formed this hybrid, which is a diploid, and then that hybrid has subsequently gone on to backcross to either parental species to produce the AAB or A-type pathogen, or the ABB or B-type pathogen. That's annoying, that's a mouthful, but this is the simplest way we can represent this. So this diploid intermediate has been formed twice. We know that from mitochondrial data, but the number of times that this backcrossing event has occurred, we don't know, or we didn't know. So these original diploids formed over here in Western Australia, right on the coast, that's Shark Bay there, really beautiful region. And the pathogens have subsequently spread all the way out across to Central Australia. So they have quite a widespread distribution. And I want to know how much clonal diversity is present in that distribution. So to do that, I sampled 257 parthenogenetic and sexual geckos. Some of those were housed in museum collections. Some of them were in universities in Australia and the States. And some of them I went and collected from the field myself, which was a pretty awesome experience. I extracted DNA and then conducted reduced representation sequencing using a modified RAD-seq method that is produced by this company in Canberra called Diversity Arrays Technology. I then did not know the exact identity of all of these samples. As I have said, some of them had karyotypes, so we knew the ploidy, and for some of them we didn't. So I was able to estimate ploidy from these reduced representation sequencing reads using a statistical program called nQuire, and I could corroborate that with my known karyotypes, and it is about 98% effective, which is amazing that you can just do this statistically from reads. I've generated a reference genome, which will just brush over all of that work there. But there's a reference genome, and I aligned the reads to the reference and called SNPs using GATK's haplotype caller program, which is agnostic to ploidy, which is wonderful. So as I mentioned, I was able to go and sample geckos from the field. So I went to some really amazing and beautiful places in Australia. This is all in Western Australia, Karajini National Park, Coral Bay, amazing, beautiful. And then I spend most of my time in these eerie and rather sad-looking rubbish dumps on station properties, because that's where it's really easy to find a high concentration of these geckos. They love to live under white goods and tin, and yeah, my colleague and I went through taking photos of these sort of in the field office setups, which is yeah, a bit of a running thing on Facebook, but it's quite interesting. So let's get into the results. So the initial SNP genotyping of the triploid parthenogens suggests that there's actually quite extensive clonal diversity in this system. We knew there was a bit of diversity because Allozyme work from 30 years ago had suggested that there was, but this amount of diversity was not necessarily expected. Don't pay too much attention to the different populations there. The important thing to note is that there is this clear divide between the parthenogens that are found in the west of Western Australia and those that are found in Central Australia. And it looks like that could be due to back-crossing having occurred across their range. And indeed, if we look at the sexual populations, we see this same east-west divide. So I thought, okay, back-crossing is widespread, the diploid intermediate might have been widespread. How can I test this in a more robust manner? So to do that, I've used this subgenome analysis technique, which was developed by Blishak and his team in 2018. And basically, I use estimates of allele frequencies in the parent that has given rise to the diploid component of the triploid genome to call SNPs in this diploid component. And I've done that for both the A and the B type parthenogens. And with that, I can basically compare the sexuals and the asexuals directly, because I can't compare diploid and triploid genotypes directly. It's like comparing apples and oranges, but this allows me to tease out, to put them into the same analysis. And what we can see is that across the landscape, we could see clustering of sexuals with asexual clones, suggesting that the back-crossing has occurred in those different regions across the landscape. So that's in the A-type parthenogens, and we also see this more broadly in the B-type parthenogens, which are found in Central Australia. So that suggests that the back-crossing is widespread. Potentially locally adapted alleles could have been introduced into the clones. That's a hypothesis, but at least we know that there's quite a wide array of diversity. And if we look at heterozygosity in the diploid component of the triploid clones, we can see that it's very, very similar to the relevant sexual. And that, once again, confirms that there has been crossing to a diploid intermediate throughout the history of these triploid parthenogens. So with that many clones present, does that explain why they've actually been able to persist for these hundreds of thousands of years? There's some degree of redundancy, there could be local adaptation, and there's the ability for clonal selection to operate on different clones. We don't know, not sure. I'm still looking at a few more things in the next year and a half of my PhD, so I want to look at more detailed sources of variation with long-read sequencing. And as you can see, there are some clones. So we have like big clouds for some of the back-cross clones. We have very tight clusters for others. I want to know whether perhaps epigenetics is responsible for facilitating adaptation to environment in these really low genetic diversity clones. So more to come, watch this space. But that's all for now. I'd just like to thank my lab and thank everyone for coming to listen. So I think we've got a couple of minutes for questions. Good. Yes.
Talk. So I'm actually just going to start with asking you to please come to the International Polyploid meeting
Yeah.
in 2026
Yeah.
in Netherlands. But other than that, because I'm one of the organizers for it, but I wanted to ask, do you think that environmental stress factors in the distribution of your polyploids and non-polyploid populations? I think there's a similar system in like Australian frogs.
Yeah. Yeah, so there is a well-known sort of documentation of a phenomenon called geographic parthenogenesis, which is that these parthenogens, and particularly the polyploid forms, tend to arise in regions that are more hostile or harsh or arid. And the time at which these parthenogens arose was a time of like great aridity in Australia. So yeah, that is often correlated with asexuality and the polyploid nature that comes with it. I'm not sure about the initial event, but of course, if it's a time of great hardship, the fact that you don't need a male and you can exist with a lower population density and still reproduce, that's highly advantageous. So that's the theory, but yeah, I'm not testing that. I think I should swap over unless there's another question. Yeah.
How important do you think somatic mutations are in the system?
I'm not too sure, because unless that was able to affect the germline, that's going to end at the end of that clone. Yeah, not too sure. But germline mutations, you'd think that like the actual mutation rate is going to be quite important, given that there's no variation introduced from recombination. But I'm not sure about somatic mutations. It's a good thing to think about. Yeah. Okay, I'll swap over. Thank you, everyone.
Okay, hi everyone. I think it's time to start. So I am Jana Flury. I am at the University of Basel in Switzerland. And today I'm also going to talk about the bees and the flowers, but more about how the flowers got independent from the bees. So in hermaphroditic plants, we have two ways of sexual reproduction. So the first way is cross-pollination, where pollen from one plant lands on the stigma of another plant, by bees, for example. But there is also self-pollination, where own pollen lands on the stigma of the same plant. And these ways come with different advantages and disadvantages. So when you outcross, so cross-pollination, you enhance genetic variation and adaptability. However, there is always the risk of not finding a mate, especially in populations that are not so dense anymore. For self-pollination, this problem does not exist, so you have reproductive assurance. However, self-pollination is kind of the most extreme way of inbreeding, so you could suffer from inbreeding depression. So many plant species have evolved a way to avoid self-pollination to kind of, also avoid these negative consequences. And one of which is the self-incompatibility locus in Brassicaceae. So this locus looks the following. It has two main genes, SRK and SCR, which are tightly linked with flanking genes like RK and UBOX. I mean linked because they always appear together, and it's a bit difficult to sequence these genes because they are highly variable. So we targeted this problem by sequencing them with long reads, so PacBio, to really get the full length of the genes and also have them like in their continuous sequence. And how does this self-incompatibility work? So when an own pollen lands on the stigma of a flower, we have the SRK expressed in the female part of the flower, so on the stigma with two alleles here, S1 and S2 of the S locus. And the pollen has, so the SCR is expressed in the pollen, but the pollen grain is haploid. However, the pollen coat is still diploid. And as soon as one allele from the S locus matches, so SCR and SRK match, the pollen will be rejected. I'll explain this a bit in more detail. So again, you see, can I use the mouse? You see the pollen corn with the diploid pollen coat and the stigma cell wall, and SRK is expressed in the stigma cell wall, so you have this protein here, and from the pollen coat you get the SCR protein. And as soon as they bind together, there is an autophosphorylation of the kinase domain, which leads to an inhibition, which in the end results in pollen not being hydrated, so the pollen tube will not grow. So all in all, these S alleles are very important for self-incompatibility. They underlie frequency-dependent selection, because imagine in an outcrossing population, you would want to have as many as possible so that they do not always come together. Therefore, we have up to 9 different S alleles in a North American Arabidopsis lyrata population, but in exclusively outcrossing species, there may sometimes up to 63 of these S alleles present. To make this even more complex, there is a dominance hierarchy of the S alleles, so one can kind of overrule another. These S alleles are also highly conserved, so you can find the same S alleles in different plant species, for example, in Arabidopsis lyrata, but also in the exclusively outcrosser Arabidopsis halleri. This breakdown of the self-incompatibility has been documented, has been seen quite often, but not well documented, or the documented cases are still quite thin. And the breakdown actually in Arabidopsis lyrata has also been documented, but the genetic reasons or what happens with the S locus is still unclear. So in our study design, we have used populations around the Great Lakes in the US, 16 in total, and we used populations that are at the distribution edge. So they form this kind of geographical groups. However, what we also tried to is, to make sure of that they are also each other's closest relative. So if you look at genes outside of the S locus, you can find this phylogeny. And for each of these groups, we have a selfing population and an outcrossing population. Really compare narrowly these independent shifts from outcrossing to selfing in these groups. And also we have two mixed mating populations in orange, where some individuals can self and some cannot. So there is a bit of theory or ideas how this breakdown can actually happen, which is there might be now a recombination between SCR and SRK, which kind of makes the self-recognition difficult. Or there can be a mutation, which leads to a loss of function of the gene, or the gene disappears completely. Or there could be modifiers that actually change the expression of the S locus, so the S locus alleles are still there, but they are modified. So for the first cause, a breakup between SRK and SCR, I want to show you. In these two populations, we find SRKs and SCRs that are always linked. So they always appear together, which is the SRK S19 and an SCR, which has not been documented so far. And in the other populations highlighted, we have the SRK S3, which always appears with SCR, Arabidopsis helleri S28. So there has not been any recombination documented between these alleles. However, we have a break between SRK and SCR in the mixed mating populations. So this is a different SCR from this, and this has even kind of switched its place, and this appears with the SRK allele S13. And further, another incident in these mixed mating populations happened so that the SRK S1, the SCR allele S1 appears with two different SRK alleles. So also there has been a break between the association of these alleles. The next cause we looked into happened in this very northern group, up 11AC and 11Z, where we could not find the SCR gene anymore in the selfing population. And this has also happened in the very south with 11Z and 11D, where we could not detect the SCR gene anymore, not even a hint. And the third cause, the modifier. So we have observed that we have the same SCR-SRK combination, but they appear in an outcrossing and also in a selfing population. So the S locus, it looks more or less exactly the same. Even these positions, they are real, so they are real base pairs where these genes were found. So it's really more or less almost a copy of each other, but we have a plant that can self and a plant that cannot self. And this pattern has also been found again in the mixed mating populations. So you have individuals here in red that do self. The orange one is mixed, so some fruit develops, some does not. And you have the outcrossers. And 11W is actually a exclusively outcrossing population next to it. And again, we have the same SRK-S1 allele with the SCR1, but in selfers and outcrossers. So what does this mean? There is probably a modifier outside of the S locus, which kind of segregates independently from the S locus and can turn the S1 allele of SRK sometimes functional and sometimes not functional. And again, the same pattern has been found in another mixed mating population, where we have again this SRK-S1, which is probably modified, but we have a second SRK allele S3.2, which is probably also underlying this modifier or another modifier. Only SRK-S13 only appears in mixed mating and outcrossing individuals, kind of giving us the idea this is still a functional S allele and it's not underlying a modifier. So to summarize this, we have found in these independent switches to selfing that in these two cases, at the very northern tip of the population and the south, there was probably a loss of SER that caused this switch. Then we have several occasions where we suspect a modifier to be in action, also on different SRK alleles. And maybe a bit of a inside note here, both S1 and S3 are lowest in the dominance hierarchy. So all other S alleles kind of overrule them, just as a little side note. And then we have this fixed selfing SRK-S19 allele, which was probably driven to fixation in this selfing population, so it's the only S allele that appears in these selfing populations. So in conclusion, there are likely several underlying molecular bases for the switch to selfing in Arabidopsis lyrata, which could be the loss of one of the genes, a modifier that is allele specific that kind of changes the expression, or a S allele that has been associated with selfing and driven to fixation. Then this breakup of linkage we saw in the mixed mating populations between SRK and SER could be a hint toward a weakened selection of this tight linkage, because there is another modifier, another gene that is actually managing the self-incompatibility. So this breakdown seems to be evolutionary labile with a number of molecular options. And interestingly, in some Arabidopsis thaliana accessions, they try to restore self-incompatibility by transferring working S-locus genes into different accessions. And what they found that it sometimes worked, but sometimes it didn't, which could be a hint towards that there are also modifiers segregating in Arabidopsis thaliana, which are changing the expression of the S-locus genes. And with this, I'm at the end of my talk, and I would like to thank my co-authors and my group in Basel, and thank you for your attention. Yes.
Do you have any idea how stable the mating systems are in these populations? This is like the outcrossing populations.
I mean, it's always assessing outcrossing and selfing. It's never 100% outcrossing. There is always some leakage that suddenly in some individuals it works. So what does this mean? Could still be that there is how much of the gene is expressed, that this is kind of varies a bit, so that some suddenly can self, but there's never a 100% never self rate in any of these outcrossing populations.
I was also just thinking about occasional gene flow from primarily self-populations.
Yeah, I mean, it's possible in, how can I say? So selfers, of course, when you have strong selfing, they immediately fertilize themselves as soon as the flower kind of opens. So there is like a small chance that actually pollen is able to cross to some outcrossing populations, but it happens at a very low rate. So it could still, of course, be that some, for example, this SRK19 allele, that this somehow ends up in an outcrossing population from time to time. But with this dominance hierarchy, if you have a very dominant outcrossing S allele, even this gene flow, can just kind of vanish due to this dominance hierarchy. Yes?
So related to that, did you look at variation in autonomous selling rates? So some of them still being pollinated, but some being self-fertilized? Is that the point?
You mean in the other regions of the genome?
No, just among populations.
Sorry.
There's variation in whether they can autonomously sell
Ah.
or whether they still need to pollinate.
No. I mean, we have taken notes for some plants. They just have fruit. We do nothing. And some you have to kind of fertilize by hand, and then they have fruits. But we did not account for this, yeah, right now. No, thanks.